Good evening, one and all. I warmly welcome you to our seventh CellTalk webinar. In today's session, Mr. Numair Arshad will be presenting research titled CRISPR-Cas knockout of miR-21 reduces glioma growth. Numair is a clinical content writer at Rnas. He is responsible for writing scientific medical white papers and blogs. His work is centered around making complex scientific concepts accessible to lay people, and he is currently pursuing PharmD at the University of Lahore. His writing at Renix focuses on precision medicine for cancer patients. Numair is currently working on gene map projects and delivering scientific content that aims to provide gene-centric information for various diseases and their treatments. Renix has also allowed him to gain leadership in various projects related to content. So without further ado, I welcome Numair to take over.
Thank you so much, Aditi, for the kind introduction. So the topic of my presentation is role of microRNAs in glioblastoma. The paper I am presenting today is titled CRISPR-Cas knockout of miR-21 reduces glioma growth. It is originally authored by Neeland and others. I took this article from Molecular Therapy Oncolytics. Its impact factor is 7.2 and the article is published in 2022. It has its major impact on the glioblastoma multiforme, but you can say that it is indirectly has some impact on the breast cancer and colon cancer as well, but it is only going for the glioblastoma multiforme. So just to start with the introduction, general background of glioblastoma. It's an aggressive form of glioma with an average survival time of 8 months. It's a very heterogeneous disease, and some of the heterogeneity can be attributed to dysregulated microRNAs. So dysregulated microRNAs is going to be the focus of this presentation. Before going into the microRNAs, I just want to introduce what they are. MicroRNAs are actually non-coding RNA molecules. They are involved in translational repression, targeted cleavage, and de-adenylation of messenger RNAs. The specific microRNA that I'm going to be talking about today is miRNA 21 or miR-21. It plays a lot of different and very important roles in biological functions, but I'm only concerned with the cell cycle regulation that it plays its part in. Expression of miR-21 has been found to be dysregulated in almost all types of cancer, and that's why it is classified as an onco-MIR. So we know it is an onco-MIR, but we want to know what is the exact role it plays in glioma. So this is the present challenge, that we want to know its exact role. And to find out its exact role, loss of function assay is required. And that has been done before, but in those studies, they use the antisense oligonucleotides. Now, antisense oligonucleotides, they have some drawbacks. The first drawback is they produce off-target effects, so they can bind to other locations in the genome and produce their effects there that in turn just alters the results of the study. Secondly, they have shorter lifetime, half-life, so an alternate method of miR-21 modulation is required. And also we need information about the specific role of genes that are being upregulated or downregulated under the influence of microRNA 21. So the objective of this research is clear. Firstly, we want to have an alternate method of miR-21 modulation that they recognized is CRISPR genome editing technology. The second one is they wanted to study the miR-21 effect on the expression of different genes, and ultimately find out what is the effect of miR-21 expression on tumor growth and spread. In the methodology, the first step is knock out miR-21. Now, miR-21, it has, the researchers used 3 different glioma cell lines The two were from mice, GL261 and CT2A, and one from human glioma cell line, that is U86. They used 5 different CRISPR RNAs for efficiency, and they ended up choosing AIR327. That was when they found out that this particular CRISPR RNA had better results in DNA messenger sequencing. CRISPR-Cas12 plasmids were used, and they had fluorescence markers on them. They used these fluorescence markers in the second step, which is to recognize, isolate, and clone the edited cells. So they recognized the edited cells using the fluorescence activated cell sorting. And secondly, when they recognized them, they isolated them and cloned them. So the significance of cloning them is that all the cells within one clone group, they have the same genome, and also they have the same editing events. Thirdly, they validated these methods by measuring the expression of miR-21 using qRT-PCR. So they took 3 different samples. First is the cell lines where they successfully knocked out the miR-21. Second is the wild type sample where they did not knock out. And the third, as control, they used the miR-21 knocked out primary neonatal astrocytes. What they found is, which was expected, that the expression of miR-21 was lowest in the knockouts, and it was not lowered in the wild types. Then in the methodology, they measured the levels of miR-21, miR-29B, and miR-15B. They found that their levels were similar between the knockout and the wild type cell lines. Then they performed NGS CRISPR-SO2 and to confirm that the knockout cell lines had significantly more modified traits percentages than the wild type cell lines. CRISPR induced insertion deletion confirmed that there were no off-target edits. So all of these 3 steps they performed to just confirm that there were no off-target effects. These are different miR-21. These are all different microRNAs from the one they are targeting. So they just confirmed that there are no off-target effects. The summary of research is that successfully edited 2 mouse and 1 human glioma genes, glioma cell lines using CRISPR genome editing technology, and they validated their methods. Next, they will observe the effect of miR-21 inhibition in cell lines and living mice models. Firstly, before measuring their effect, what they did is they measured the expression level of VMP1 gene. So why they did is this step is miR-21 is actually located within the intron of this gene VMP1. So when we are editing miR-21, it is expected that the VMP1 might get altered. So to confirm that no such off-target effect is happening, they performed 3 different tests. The first is qRT-PCR. And here you can see that their amplicons of the wild type and knockout, they are very similar. They are comparable. This tells us that the level of VMP1 is comparable between the wild type and knockout cell samples. The second test is a Western blot analysis. And here you can see that they compared the wild type with the different cells from the clones that they made. And the mark is very similar, and at least in these 2, and you can see that the VMP1 protein is being expressed in these wild types, also in the knockouts. So they also compared it with the housekeeping protein, and this also tells us that the protein level of VMP1 is not being affected by the inhibition of miR-21. Third test is immunohistochemistry assay. And here you can see the red color. This red color represents the presence of VMP1 in the knockout samples. So this is another test. They use these three tests to just confirm that VMP1 is not being altered. After that, they moved on to measuring the exact gene expression levels that are being altered by miR-21 inhibition. So they found that in total, there were 52 genes that were being affected. 25 of them were being upregulated and 27 were being downregulated. Here you can see in the MA plot, these red dots, they indicate the each one red dot is indicating one gene that is being affected by miR-21 inhibition. It is better represented here in heat map. Here you can see that this heat map actually has two portions. One is for the wild type and second is for the knockout samples. Here you can see that some of the genes that they are being underexpressed when miR-21 is present, but they are being expressed more amount when the miR-21 is not present. Out of all these genes, you really have to focus on these two, CDC25A and CXCL10. So both of these genes are being less expressed in the wild type samples and they are more expressed in the knockout samples. So this means in the presence of miR-21, their expression is reduced. Next is they just reintroduced the miR-21 in some of the wild type samples, in some of the knockout samples. And for this purpose, they used the lentiviral vectors. And as expected, when they reintroduced these miR-21, their expression level jumped back to normal level, which was compared to the wild type. After reintroducing, they went back and performed the same expression level of different genes. These are some of the important genes. Previously, we saw that 52 genes were being affected, but here they just focused more on the very important genes. So these are important genes and they measured their levels. And what they found is in the wild type, their expression is low as shown earlier. In the knockout, their expression is higher. But here when they reintroduced miR-21, their expression fell again. So this really goes on to tell us that it is really miR-21 that is inhibiting their expression. So this is another validatory step that they performed, and it really confirmed that miR-21 is reducing the expression of these genes. You can see the same pattern repeated over and over for all of these different genes. Third, they measured the effect of miR-21 inhibition in vitro proliferation, migration, and invasion. What they found is when the level of miR-21 was low, the proliferation rate was also low. So same thing here, it is shown in both these cell lines. First is for CT2A cell line, its wild type has a more proliferation rate, and then its knockout variant, and same thing is repeated in this one, U87. And they performed the same experiment with the third cell line, that is GL261, but here they also taken into account the restored variants. So you can see that the proliferation rate of wild type and restored is very close to each other, but this knocked out version, it has very low proliferation rate. So previously we saw the graphs. These are the actual images. You can see in this image that the wild type has more proliferation rate as compared to the knockout ones. This is the comparison between the wild type and knockout. You can see the same pattern repeating over and over again. And in here they went one step ahead, and they measured. They also compared it with the restored versions, where the miR-21 was restored using the lentiviral vectors. And here again, you can see that in the wild type, the proliferation rate is higher than the knockout, but when they restored, the proliferation rate jumped back on. The same pattern can be seen repeating itself. The third step is that they measure these effects on the proliferation, migration, and invasion in the living mice. So for this purpose, they injected the knockout and wild types to mice. They actually took two groups of mice. For one group, they injected the knockout glioma cells where the miR-21 was not being expressed, or it was expressed in very low amount. In the second group, they injected the wild type cells, glioma cells. And one very obvious thing that came out of this is the wild type group of mice, it died very early as compared to the knockout group. So you can see here, for example, in this GL261, wild type died after 21 days, but the knockout lived for at least 60 days. It actually didn't die after 60 days, but they had to sacrifice it to get the brain slices, because they needed to look for the presence of tumor in its brain slices. So it at least lived for 60 days, and similar pattern can be seen in other cell lines. Then they performed a representative in vivo imaging system images, this analysis, and you can see the tumor is encircled by this red line. It is repeating itself. The same pattern can be seen. You can observe the glioma is present in all of these. These two actually died, so their result was not taken. But in the knockout, you can see that the glioma is not very apparent. It is apparent here, but in rest of the images, it is not apparent. So you can see a clear difference between the wild type and between those mouse which were injected with the glioma where miR-21 level was reduced. So this again tells us that the proliferation was reduced when miR-21 level was reduced. And finally, they took their brain slices and they stained it with DIPI and GFP. And here you can see all these images on top are for the wild type where the level of miR-21 is high, and these bottom images are for the knockout sample where the miR-21 expression is very low. And the glioma here is encircled by this dotted line. You can see it is very apparent in the wild type, but it is not apparent in the knockout group. In the discussion, they first measured the level of miR-21, and this showed us that there were no off-target effects, and it was really the translation and maturation that was being affected by this gene editing, and transcription was not affected. This was again confirmed because they found the level of miR-21 intact in the knockout samples. Secondly, they measured the level of VMP1 in the knockout samples using 3 different tests that we discussed earlier. After that, they looked into the specific CDC25 genes. These are the specific that we are going to discuss. First gene, CDC25A, it is generally regarded as pro-oncogene, that it promotes tumor, but here we found, contrary to the literature, that the tumor actually reduced when CDC25A was being expressed in more amount. So one possible explanation of this can be the abundance of CDC25A after miR-21 knockout results in nuclear localization, and increase in apoptosis in glioma cell lines. Similarly, they found the same pattern with CDK6, and it is also considered a tumor promoting gene, but here they found that its upregulation actually inhibited the tumor growth. So possible explanation can be post-translational summarization. Again, from these two genes, CDC25A and CDK6, they tell us that it is really the post-translational modifications that are determining what effect these genes will ultimately make on the tumor inhibition or tumor promotion. And these post-translational modifications are in turn being dependent on the amount of these or the expression of these genes within the cell, and expression in turn is being dictated by the miR-21. So in other words, miR-21 is increasing or decreasing their level in the cell, and their level are determining what sort of post-translational modifications they will undergo, and those post-translational modifications are determining whether they will cause the tumor or they will inhibit the tumor. Then they also discussed the CXCL10 microRNAs, and this was actually in agreement with the literature. Next, SOX2 gene expression was also taken into consideration, and it is actually reported as one target of miR-21 in the previous literature. But here they did not find its level being changing in response to inhibition of miR-21. One possible explanation of this can be that in glioma cells, SOX gene is being expressed so much by all sorts of other means that the knocking out miR-21 did not produce any observable results. That's why it was not included in the 52 genes that were being affected by inhibition of miR-21. Now, to conclude, the authors of this paper successfully knocked out miR-21 in glioma using CRISPR technique. This resulted in decreased proliferation, migration, and invasion of these cells in vitro, as well as increased the survival of tumor-bearing mice in vivo. So the impact of this research is, firstly, it contributes to the general RNA therapy that is being practiced in cancer. Secondly, there are clinical trials that are already going on. So these clinical trials are looking into the inhibition of miR-21 in other cancers, not the gliomas, not the glioblastoma, but in other human cancers. But this study emphasizes that it is important to just understand the cellular mechanisms and cellular targets of miR-21 inhibition before moving on to exploring its therapeutic benefits. So, for example, we saw that CDC25A, it is generally regarded as pro-tumor gene, pro-proliferative gene, but in glioma, it was actually inhibiting the tumor. So before moving on to proceeding to exploring its therapeutic benefits, we should really check for its specific roles in glioma cells. Thank you so much.
Thank you so much, Umair. That was a truly illuminating and educational talk. I'm sure we've all had a lot of takeaways from this webinar. I will now open the floor to the audience for any questions, concerns, or opinions that you all have for Umair. I can see 2 questions in the chat box. The first one is from Nupur. She asks that how CDK6 and CDC25 genes are related with miR-21.
So in wider literature, they are regarded as the genes which are being promoting the tumor. So whenever miR-21 is expressed in greater number, they are also expressed in greater number. So they promote the tumor. But in the glioma, the researchers found different to that, and that really emphasizes that before just targeting these cellular mechanisms, we should understand them in each individual cancer. Like they were promoting cancer in other human cancers, but here they are inhibiting the cancer. So it really emphasizes that we should understand their specific roles in glioma before going on to exploring their therapeutic benefits.
So we can see that knocking out miR-21 affects multiple genes.
Can you repeat that?
So can we say that knocking out miR-21 affects multiple genes in glioma? It targets multiple genes.
It does. In total, they found 52 genes that were being affected by knocking out.
Yeah, that's the good thing or good thing of this study, I think.
Yeah.
Thank you. Nice talk.
Thank you.
So our next question is from Vijay. What approaches do the authors suggest for genetic modification of solid GBM tumors for miR-21 inhibition? He asks.
They did not discuss about that, the solid tumors. They were all these other solid tumors. They were only talking about glioblastoma, and it was really a preclinical study, so they did not go into the delivery of these approaches, but they really wanted to emphasize that the mechanisms that were behind this. So they found out what genes were being affected, what was the effect on the in vivo proliferation, what was in vitro. So they did not really talk about the delivery of it. But if you search the wider literature out there, you can find out that currently other solid tumors that are being targeted, they are using the exomes. They are incorporating the, for example, one chemotherapeutic drug in the exome, and also they are adding this oligonucleotides in that. So they are only using the oligonucleotides. They are not using the CRISPR so far, and for that they are using the exomes.
I hope that answers your question, Vijay. So thank you so much, Numair, for that webinar. It was very helpful and very educational. So while closing this webinar, I would also like to inform the audience that the next CellTalk webinar will be held on the 12th of August, 2022. And Mr. Jivir Kapoor will be presenting on the topic, comparing the average cost of outpatient care for public and for-profit private providers in India. I hope to see you all next week, this very time. And thank you so much for your presence. Have a nice day.